Hello, everyone, and welcome to today's live broadcast, Growing Together, Lessons Learned from the Characterization of the Pediatric Microbiome, presented by Dr. Emily Hollister. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Roche Diagnostics. As a global leader in healthcare, Roche Diagnostics offers a broad portfolio of tools that help healthcare providers in early detection, prevention, diagnosis, and treatment of diseases. In molecular diagnostics, the company is driven by a vision of working with laboratories like yours to improve the medical value you offer in microbiology, infectious diseases, oncology, and genomics. Roche Diagnostics continues to meet unmet needs through its investment in research, innovation, and scientific excellence with the goal of supporting your important role in improving patients' lives. To learn more about Roche Diagnostics, please visit usdiagnostics.roche.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Dr. Emily Hollister. Dr. Hollister is a microbial ecologist and bioinformaticist. She's interested in quantifying and characterizing microbial communities from a wide variety of environments, including the human body. Utilizing high-throughput next-generation DNA and RNA sequence approaches, her research seeks to improve our understanding of microbial communities, their distribution across time and space, the ways in which they function, and their influence at broader scales. Dr. Hollister currently serves as the Director of Microbial Ecology and Bioinformatics for the Texas Children's Microbiome Center. Please join me in welcoming Dr. Hollister. I will now turn the presentation over to her.
Thank you, Judy. Before I get started, I'd like to disclose that this is a sponsored talk by Roche, and with that, we'll launch into my presentation, which, as Judy mentioned, is entitled Growing Together, Lessons Learned from the Characterization of the Pediatric Microbiome. Today, I plan to discuss with you the importance of the pediatric microbiome, the gut microbiome and its relationship with human development and the aging process, and some other application of microbiome science in pediatrics. Over the course of the last 10 years, microbiome-related studies have gained tremendous foothold in the literature and popular press. This has been fueled by advances in sequencing technology and a growing awareness of our microbial world. As such, the science has begun to describe microbiomes in places we never thought to look or tried to look before. And it doesn't take much work beyond the pages of Cell Science or Nature, or honestly many journals beyond that scope, to find that there's no end to the variety of microbiome studies being conducted, from describing the structure and function of the healthy human microbiome in the context of the Human Microbiome Project sponsored by the National Institutes of Health here in the United States, to looking at the gut microbiome across age and biogeography, to looking at the microbiome in a variety of other locations and places. New microbiome findings are being reported on a daily basis. And what are we learning from all of this? It's been about 10 years since the first articles describing next-gen sequencing were published, and about 8 years since the launch of the Human Microbiome Project. And really what we've learned from this to date, it has been in large part, natural history. A large amount of time and effort has gone into documenting what occurs when and where, And it's led to helping us define, or at least begin to define, healthy and normal for a variety of community types. Very interestingly, and certainly of importance to my own field, defining or linking changes in microbiome to states of disease and health. And as we think toward the future, rethinking conventional approaches to management, therapy, diagnostics, and treatment. We know that the human gut microbiome is intimately linked to our developmental continuum. And this conceptual diagram from a Nature Reviews Microbiology manuscript published about 4 years ago provides a conceptual outline of these microbiome changes over the course of one's life and the various factors that influence microbiome structure and function over time. As we move through various life stages, a variety of factors contribute to shaping our microbial communities. This may be factors such as host genetics, delivery mode, and diet in early life, the developing immune system, illness, nutrition, antibiotic exposure during childhood, and a variety of other things as we move through our life stages. Exposure to different people, compounds, substances, diets, stressors, a variety of things interact with our own bodies and our microbiomes to shape their interaction with one another. Although there are exceptions, most of the microbiome studies to date have focused primarily on infants or adults. And with studies like the Human Microbiome Project and MetaHit providing large amounts of information regarding the healthy Western adult microbiome, also providing some insights into IBD. The pediatric microbiome, however, especially in children and teens, is less well characterized. Being at a children's hospital, this has been a main source of work and a focus for us in the last couple of years. But why is the pediatric microbiome so important? Well, a lot of research suggests, and a lot of thinking suggests, that the pediatric microbiome has the potential to influence the shape of things to come. We realize that there is a growing awareness in the community of the importance of the microbiome during pediatric development and its potential to influence health outcomes later in life. And evidence for this is mounting. It appears, and there certainly appears to be a link between the gut microbiome and autoimmune status, and work from a couple of different publications over the last couple of years have demonstrated this, either from the perspective of priming the immune system early in life, or signaling autoimmune changes at the onset of type 1 diabetes, for example. We also know that the pediatric window and the gut microbiome are linked to cognitive development and behavior, and there's been some beautiful work done in animal models suggesting that cognitive development and behavior are very closely linked to the microbiome. Work in germ-free animals, highlighted here on the left, has demonstrated that germ-free animals, or those lacking an intact microbiome, showed decreased inhibition, and that there's a specific window in which this can be corrected. Now, decreased inhibition is a hallmark of abnormal behavior in mice, and so mice lacking an intact microbiome behave differently from those who are raised under conventional conditions. Likewise, recent work published in Brain Behavior and Immunity suggests that alteration of the microbiome with chronic antibiotic exposure can also lead to altered behavior and diminished cognitive ability in young mice. In both cases here, the lighter color in each figure represents a control mice, and the darker color represents microbiome depleted or altered, i.e. germ-free or antibiotic-treated mice. So clearly there's a strong link between the microbiome, or at least evidence is suggesting strong links between the gut microbiome and our own development and behavior along this gut-brain axis. We know that the animal models also suggest that early life antibiotic exposure and modulation of the microbiome can also lead to increased risk for obesity and adiposity. Work out of Marty Blazer's lab in New York and work performed by Lori Cox and others suggests that chronic early life antibiotic exposure can lead to a disrupted microbiota and the potential for increased adiposity. So as we think about this, the human microbiome and the developmental continuum, we're seeing mounting evidence from both human studies and animal models suggesting the importance of the microbiome during early life. I'd like to invite you to take a step back, though, and consider what else we know about the microbiome and our sort of earliest stages. So we've learned over the last several years that we're not necessarily sterile at birth. This has been a long-held assumption that a newborn or the fetus is more or less protected from microbial exposure until birth. However, a variety of studies have demonstrated that meconium, or basically the first fecal material, actually is fully populated with microbes at the time of birth. What I'm showing you is microbial community characterization from meconium performed or published in 2013, where each one of these pairs of bars represents a single infant sampled at different points in the meconium, either an internal sample or an external sample. And what we see is that regardless of sampling point, there are detectable microbes, and there are a variety of microbial configurations, even from the first day of life. So we know that we're not sterile at birth. We've also learned that the placenta is not sterile either. This is work that was performed by Shirsti Ayer's group at Baylor College of Medicine and published last year in Science Translational Medicine. What their group found was that placental tissues harbor detectable bacterial DNA, and that the composition of these placental microbial communities is actually quite different from that of the vagina and other body sites that have been previously characterized by groups working with the Human Microbiome Project. Interestingly, this community shares a high degree of similarity, or at least when compared to other body sites, shares the greatest degree of similarity with those from oral communities, providing extra evidence to suggest the strong link between oral health and birth outcomes. We've also recently learned that the uterus isn't sterile either. This is data that appeared recently in a Peer Journal Preprints article evaluating bacterial community composition in endometrial samples from 19 non-pregnant women. And very interestingly, we find taxa that are very commonly associated with the gut, Bacteroides, Xylena solvens, as well as taxa that are commonly found in the vaginal tract, the Lactobacillus type species. We've also known or have learned that delivery mode may influence your microbiome, and conflicting evidence suggests that one's early microbiome can be further shaped beyond what's initially seeded by mode of delivery. Gloria Dominguez-Bello's group published this paper in early 2010. It's highly cited, and this group found that infants delivered by cesarean section harbor a microbiome that resembles that of human skin, while those who are delivered via vaginal mode of birth tend to represent mom's vagina. However, others who have attempted to replicate this study have found that mode of birth doesn't really matter quite so much as previously thought. Regardless of your mode of birth or mode of delivery, the human gut microbiome changes rapidly and dramatically over the first few years of life and is influenced by a number of factors, including diet, illness, antibiotic exposure. This is from a study published out of Ruth Lay's group, this Koenig paper from 2011, where they tracked a single infant over the course of roughly 2.5 to 3 years. And from birth through early childhood, through exposure of table foods, antibiotics, socialization, and other features of this child's early life, we can see that the gut microbiome changes over time. More interestingly, or as interestingly, this progression has often been described as one that's very chaotic. But in contrast, what we see is that a steady progression of an increase in phylogenetic diversity over time and an increased or a steady progression of differentiation over time as this particular child's microbiome matures. We find that steady increasing richness, or that increasing richness is a steady feature over the first several years of life. This is data published by Tanya Yatsenenko in 2012. And others have found that this may actually serve as an index for maturation. When we look at children who are developing normally versus those who may be malnourished and falling off of the regular growth charts, we find that the steady progression tends to be a consistent feature if one is healthy. But if one tends to be falling off of this track, it could suggest that the microbiome is serving as an indicator of malnutrition and poor development. As we continue to mature, the gut microbiome becomes more adult-like over time, suggesting that by the time a child reaches roughly 3 to 4 years of age, their gut microbiome has reached an adult-like stage and sharing roughly 50 to 60% similarity with those of adults, or older children and adults. But as notably, the microbiome continues to change as we age, and not surprisingly, really this maturation doesn't end at the age of 3. We find that the microbiome continues to change over time, regardless of where you live or which culture you belong to, and that even into adulthood, one can use microbiome-based markers to distinguish younger adults from older adults and centenarians. And this change involves both composition, so the types of bacteria that are present, as well as function. When we compare adults and infants, not surprisingly, we find very dramatic differences in function and functional potential of the microbiome related to things like amino acid metabolism and utilization of vitamins and cofactors. If we take that same type of functional information from a shotgun metagenome, we found, or Lan et al. Actually found, that one can reasonably distinguish younger adults from older adults, suggesting that there is a young adult signature and an older adult signature that can be picked out from functional information of the gut microbiome. Taking a couple steps back down that developmental continuum, we find that even though children can be distinct, or children tend to reach an adult-like stage by the time they're 3 or 4 years old, children and adults still differ from one another quite dramatically with respect to their community composition and the types of functional genes that they carry. And this is consistent across multiple sequencing technologies. What I'm highlighting here is data that we've recently published out of our group at Texas Children's and the Texas Children's Microbiome Center, where we compared healthy adults and healthy children with one another. These are adults who were recruited as a part of the Human Microbiome Project and happened to be from the Houston area, and healthy children who were also recruited from Houston, Texas. Utilizing similar sequencing approaches, we characterized communities using 16S ribosomal RNA gene sequencing as well as shotgun metagenomic approaches. And what we find is that regardless of sequencing approach, we find relatively consistent differences in community composition. Now, we're not the first to have described this. Others have looked, a few others have looked at comparisons of children and adults using microbial community composition, and not surprisingly, when they do look, they do find differences. Whether they used 16S sequencing or microarray technology, we find that children and adults differ from one another with respect to composition. However, our group is among the first to have taken the next stage to carry this out and look at functional differences. And what we find is that in combination with these taxonomic differences, we do see some differences in functional potential between children and adults. These build upon those that have been described in comparison of infants and adults. For example, Tanya Yatsenenko et al. In their 2012 Agent Biogeography paper described specific differences between the gut microbial communities of children and adults with respect to dietary acquisition versus de novo metabolism of B vitamins, including folate and vitamin B12. Very interestingly, what they reported was that adults tended to have a greater capacity for B12 metabolism than did infants, whereas what we find is that children actually exceed the functional potential for B12 metabolism or have a greater potential for B12 metabolism than adults, and adults sit in the middle of this spectrum relative to infants. So what we find is that it appears there is sort of a peak of potential in children, and correlating evidence from the literature suggests that lifetime vitamin B12 actually peaks, or blood concentrations of B12 peak around 7 to 8 years of age. So it makes sense that cooperation from the microbiome at this stage of life would go along with this. And what I'm showing you here on the left-hand side is a portion of the vitamin B12 synthesis pathway, where boxes highlighted in green are enzymes for which we see increased relative abundance of genes in the gut microbiota, or in the gut microbiomes of children relative to adults. Those highlighted in gray are enriched in adults relative to children. If we look at a broader perspective, we see that although not quite as pretty as the heat map produced by Yatsenenko et al.'s group, we do see that there is a differentiation of children and adults with respect to multiple functional pathways in the gut microbiome, again, linked to things like amino acid metabolism, excuse me while I use the pointer, amino acid metabolism, vitamin synthesis, and a variety of other types of functions. Interestingly, too, we find that key pathways that have been previously described in the context of inflammation, aging, and the development of metabolic diseases differ between children and adults. On a large scale, overall, we see that children and adults do generally differ from one another, though not exclusively, with respect to overall community composition and their functional gene pathways. But when we look at key pathways related to either health promotion or metabolic disease and inflammation, we see key differences. The porphyrin and chlorophyll metabolism pathway, again, is that vitamin B12 I just highlighted. Methane metabolism has also been linked to improved relative health with respect to the gut microbiome. In contrast, things like functions like genes involved in the carboxylic acid cycle, lipopolysaccharide biosynthesis, and oxidative phosphorylation suggest increased potential for inflammation or the ability to deal with oxidative products. And overall, these changes like these appear to have implications for health. Others have previously reported shifts in microbial composition related to general health in the elderly, for example, community richness or gene richness in the community, or total gene counts, where increased gene counts tended to be indicative of better overall health, whereas lower gene counts tend to be indicative of poorer overall health, as well as increased community richness over time. Likewise, Le Chatelier et al. Published in 2013 this high gene count, low gene count paradigm, where they described individuals who had high gene counts being of better overall health and decreased risk for adiposity and metabolic disease relative to those who carried low gene count microbiome phenotypes. So clearly, mounting evidence suggests that the microbiome does influence the shape of things to come. As we consider our knowledge of the human microbiome and the developmental continuum, it becomes apparent that recent findings are redefining our understanding of various life stages. For example, until recently, it was very much held that the unborn child was sterile, and we know now that that's not the case. That while microbial loads may potentially be very low, they are present, and we assume beginning to influence the early stages and the early foundations of the human microbiome relationship. We're also learning that once a child reaches roughly 3 years of age, there's still additional development that will go on. Not surprisingly, our bodies continue to develop over time. It appears that the microbiome develops along with us. However, we have a number of gaps in our knowledge still. Very little is known about the teenage microbiome. What happens in that complex, crazy roller coaster ride of puberty and the onset of adult sex hormones? And we're still learning more about the transition across these stages and into older age. While we do have some great baseline data, there's still a lot left to be learned. So, in addition to describing the development of the healthy human microbiome, there's tremendous interest in applying microbiome science to the study of pediatric disease. I'm going to take an opportunity here to highlight some of the projects that we've had ongoing at Texas Children's Hospital and Texas Children's Microbiome Center, where we have a very active research community who's interested in applying microbiome science to their own particular subspecialties and conditions of interest. And so I'd like to tell you a few short stories about some of the work that we've been doing. We have a couple of collaborators from the Department of Neonatology who've been very interested in studying the airway microbiome in premature infants and the degree to which the airway microbiome can be used to predict the development of serious conditions like bronchopulmonary dysplasia. This work was led by Pablo Lohman, and what we found is actually really interesting. Again, going along with that idea of the child is not sterile at birth, previous studies have attempted to address the airway microbiome in preterm neonates, and the one previous study that comes to mind actually was unable to detect bacterial DNA until 2 to 3 days of life, whereas in our study, the children Pablo recruited, we were able to get detectable bacterial DNA from birth and follow these children over time, including the development of bronchopulmonary dysplasia, or those preterm neonates who were ventilated but went on to have healthier outcomes. And what we find is that in these preterm neonates, from very early on, those who go on to develop BPD have lower bacterial richness over time, at onset and over time, and their community shifts very rapidly to one that contains taxa that are potentially problematic. So even from 3 days of life, we see that we may have a biomarker or a potential signal of things that are about to change. Beyond the respiratory tract, we will also have a variety of GI collaborators who are interested in characterizing the microbiome with respect to conditions of interest. One of these happens to be the fecal microbiome in patients with short bowel syndrome. This was work carried out by Zev Davidovics. who was a fellow here and is now at Connecticut. And Zev spent some time characterizing the fecal microbiome in a handful, well, variety of patients with short bowel syndrome, and trying to figure out the degree to which symptoms might be correlated with microbiome composition. Largely what we found was that symptoms were not, but that we could very clearly distinguish the healthy microbiome from one of short bowel syndrome. Other work that we've had ongoing has been looking at dietary interventions. In particular, Bruno Ciampatazzi from our GI division has been interested in looking at children with irritable bowel syndrome and the degree to which dietary interventions like the low FODMAPs diet may be efficacious and have an interaction with the microbiome. Early work from Bruno suggests that baseline microbiome composition may be an indicator of how well particular children may respond to this dietary intervention, which is fairly harsh, the removal of a large variety of potentially fermentable carbohydrate sources. And if we can help direct patients or predict which patients may respond better to the FODMAPs intervention before they go on it, we could save families a lot of time, trouble, and potential disappointment if the intervention didn't work. We've also been working very closely with Richard Kellermeyer in our GI division, who's been doing work with fecal microbiome transplantation. We have a fecal microbiome transplantation program in effect for Clostridium difficile infection, and we've had some ongoing work with an IND approval from the FDA looking at FMT applications in ulcerative colitis. And what Richard has been able to show is that FMT in ulcerative colitis patients does appear, at least in those with low-grade inflammation, low-grade disease, does appear to produce transient beneficial effects that from the time of enrollment, this is a case study of a single subject, from the time of enrollment through multiple transplants, we see that the microbial community changes over time, becomes much more like the donor, and following, the donor here in red, and following removal from the protocol begins to drift away from the donor and this potentially healthier state. So again, that transient effect. If we look at 3 different patients, what we find is that over time, going from an untreated state to pre-FMT to post-FMT, if we look at colonic biopsy specimens, mucosal biopsy specimens, we see an improvement in structure, in epithelial structure and overall tissue health. Providing new insight, and so we've been doing a fair amount of work to try to track microbial community response in these patients to the FMT treatment, but also track host response in terms of clinical symptoms, but also host gene expression and things along those lines. So what we find with our human microbiome work and our pediatric work is that the human microbiome in the pediatric window is definitely an important thing, and clearly provides a number of opportunities for research. We know that the microbiome, or we're beginning to appreciate that the microbiome appears to be seated well before birth and continues to develop with us as we move along the aging continuum. There's actually a really fantastic paper that was just published in Cell Reports. It's actually an uncorrected, or maybe a corrected proof at this point, from Clark et al., looking at the development of the GI microbiome with age in Drosophila, and looking at the effects of aging in the microbiome in Drosophila, particularly with respect to declines in barrier function and immune function as the Drosophila age. And this is, I think, harbinger of things to come, and as we begin to understand how the microbiome changes along with us and what the implications are for health, certainly what the implications are for health, and where there may be potential opportunities for diagnosis or intervention or potential correction or modulation as we go. clearly, growing evidence suggests that the pediatric window pediatric period represents an important window with respect to host microbiome interaction, be it the development of autoimmune disease, predisposition to metabolic disease or adiposity later in life, or shaping our cognitive development and behavior. The pediatric window appears to be very tightly linked to the structure and function of the gut microbiome, and is something that we will certainly be wanting to learn more about, be working more on, but it suggests that this is a very important period in time for our development and that of our microbiome. We're learning that the pediatric microbiome, although adult-like, is not fully adult. A child's microbiome appears to share many adult-like qualities, but differences in composition and function suggest that additional development is likely to occur. As we begin to fill in more of the blanks along the developmental continuum, this idea is likely to be fleshed out more, and the importance of it will be fleshed out more. And as I've indicated, there's still many unanswered questions with respect to the pediatric microbiome at all body sites, but new findings continue to challenge long-held paradigms. And for example, the sterility of the microbiome at birth, as well as this idea that by the time one reaches 3 years of age, we're pretty much done developing our microbiomes. So challenging long-held paradigms, informing future diagnostics. There have been some very nice applications of this suggesting that we may have some developmental indices available to us as a readout from the human microbiome. But beyond that, we may also be able to gather insight into the microbial diseases and polymicrobial diseases from the microbiome, and certainly shaping interventions. If we can direct therapies more specifically, we may, one hopes, have more success or greater success at treating disease and promoting health. So with that, I will give some acknowledgements to a variety of my collaborators based here at Texas Children's Hospital and Baylor College of Medicine, particularly those who are involved in the Texas Children's Microbiome Center, including our director, Jim Versalovic, Ruth Ann Luna, who directs our sequencing group, Sridevi Devaraj and Tor Savage, who are associate directors, and a variety of others without whom the work I've shared today would not have been possible. I've mentioned a variety of our other collaborators from neonatology, the GI and hepatology group, and we also work very closely with the Bioinformatics Research Laboratory at Baylor College of Medicine, the ALCAC Center for Microbiome and Metagenomic Research, and Human Genome Sequencing Center, and finally, of course, some of the work that I've described today was funded by the National Institutes of Health and IDDK, and specifically under this UH2, UH3 funding mechanism. With that, I think we can move on to questions. Thank you.
Thank you, Dr. Hollister, for that informative presentation. It's time for Q&A. So if you have a question you'd like to ask Dr. Hollister, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Okay, let's get started. Your first question is, can the pediatric microbiome affect a child in the most current studies of ADD, ADH, and defiant disorder?
That's a great question. There's certainly ongoing work in our group and others attempting to address this question. Or at least, well, actually, I take that back. I'm thinking more of autism spectrum disorders, but certainly some of the to my knowledge, some of these may affect or may also fall onto the autism spectrum. It's not unreasonable to suggest that they could. To my knowledge, I don't know specifically if there are ongoing studies. Work from animal models does suggest that our gut microbial communities, again, as I've showed some evidence from earlier today, can affect behavior, and it certainly may be possible. The results from the germ-free mouse models showing lack of inhibition are intriguing and suggestive, and I think this is probably an opportunity for work down the line.
Is it correct to say that vaccination can disrupt the natural process and development of the human microbiome?
This is also a great question. I do not know the answer. I've certainly seen some preprint manuscripts that attempt to address this question, though it's hard to say. We certainly know there are benefits from vaccination, global benefits, community benefits. I don't know. I think I'll leave the answer at that.
We know that we have an important bacterial flora in different organs, including our mouth. Are the bacteria that we consider useless or harmful important for the survival of our own flora, and also would our flora survive or be the same without them?
I think that the question is absolutely correct in that we have flora or bacteria reside in and on our bodies at a variety of sites, and I think that there's still a lot we're learning about the interactions among bacterial community members. So those that may not have an apparent use, and in that they're not a known beneficial organism or a known pathogen, I think there's still a lot of taxa out there that we're still just not sure what they do. We know that in some cases there is a very intimate relationship between some taxa in that they require one another for co-metabolism, for survival, for co-culture conditions, if you will. And so I think that there's still a lot to learn as to how and why those particular taxa, the ones that we think are sort of neutral, what they're doing there. And so I wouldn't, I would suggest that the author of this question is probably correct in that these particular taxa that we don't yet understand may be important for the survival of others and the ones that we tend to value.
Does an external factor, such as a family pet, affect a child's microbiome?
There is evidence to suggest that the presence of a family pet can affect the microbiome. I believe that Rob Knight's group published a paper on this not that long ago, and there have been suggestions from the allergy and immunity literature that literature and even folklore, that having a pet around can affect a microbiome and allergic responses later in life. But yes, I believe the answer is yes, that a child's microbiome can be affected by the presence of a family pet. I recall from that study from Rob Knight's group that family pets and their owners tended to share more bacterial taxa in common with one another than did two separate humans who did not live together. So the close relationship between humans and their pets suggests that there is some degree, and maybe more than we realize, of bacterial swapping going back and forth.
The vagina has bacteria, so how can a newborn be sterile?
Right, that's a great question. And so the assumption, I think the long-held assumption was that that it would be an internally sterile condition, that the lungs, the GI tract, other body sites, I guess internal body sites, would be sterile. Clearly that's not the case, and we're learning that. That, as I said, the long-held assumption in medicine, and perhaps a very false one, was that, so not so much the newborn, but the fetus, if you will, in cases of a healthy child, one that was not affected, for example, by a condition like chorioamnionitis, would be sterile. As I said, we're learning that that's not the case, and we probably shouldn't have expected that to be the case at all.
We're almost out of time, so this may have to be our last question. Is it possible to establish community types in children? Are they totally different to the enterotypes A, B, C, D in adults?
I believe people have looked for enterotypes in children, and I know early on in our group we did do some initial characterization to see if we could recreate the enterotypes described by the European authors, the meta-hit authors. So they do appear to exist. We do know that full gradient of community types, if you consider the sort of Bacteroides to Firmicutes gradient, it's present in adults and present in children, and we believe that whether you consider enterotypes or enterogradients, that full spectrum does exist in children too.
Okay, I have time for another one for you. As we know more about the human microbiome, the definition of proper personal hygiene seems to be no longer an elimination of all bacteria to stay healthy. What are the best practices to teach our children on proper personal hygiene with diversified microbiome in mind?
This is a great question. Thank you. So clearly, we are learning that not all bacteria are bad, and that we probably don't need to eliminate all bacteria from our lives, as the war on bacteria and antibiotics and antimicrobials might have led us to believe. You know, and what do we teach our children in terms of proper personal hygiene? You know, I think there's still, I think it's important to teach children that we don't necessarily need to eliminate all bacteria from our lives, that there are beneficial ones, but things like proper hand hygiene are still very important. Proper food handling techniques are still very important, because we do appreciate that although we want to keep the good ones around, we still need to be very careful about the pathogens and foodborne illness and things along those lines. So I think this is something that's going to continue to evolve. We may reach a point where, you know, dousing our kids in hand sanitizer is not the accepted norm anymore, but at the same time, using the soap and water and maybe limiting antibiotics to cases where they're absolutely necessary, utilizing more traditional approaches rather than having triclosan or antimicrobials in every personal care product we use, that we tend to stick to sort of the basics of the soap and water and less, shall we say, advanced personal care products. So I think this is a great space to keep an eye on as we continue to learn more about our own human microbiome and the ways we affect our microbiome, the way our built environment affects us, the way we interact with it. So I think that, as I said, in terms of best practices for our children, we still need to teach them about proper avoidance of pathogens, but at the same time, that important nutrition, whole foods, probiotic type bacteria, eating foods that could benefit our bodies is still an important thing to do.
hope you have time for another one. Why is it said that our normal skin flora are opportunistic bacteria?
And I see that we've had one additional question. Judy, if we have time, if we don't have time, let me know. This last question, or this last question that's come in, asks, why is it said that our normal skin flora are opportunistic bacteria? Opportunistic bacteria may not quite be the appropriate word we want to use, but certainly skin flora do contain a number of opportunistic pathogens. You can think of an opportunistic pathogen as something akin to a weed. A plant, so a weed is a plant that exists in a place where it's not appreciated. An opportunistic pathogen is also functions along the same lines, where it's present in when present in one particular location, it can be relatively harmless and a part of the normal background, but when introduced to, say, a wound or to an immunocompromised individual, can cause problems. And so there are special bacteria that do kind of have that dual role of being generally harmless in most situations, but when given the opportunity to take advantage of a situation, can be considered opportunistic pathogens.
I would like to once again thank Dr. Hollister for her presentation. Do you have any final comments, Dr. Hollister, that you'd like to share?
Well, I'd like to thank everyone who attended today for your time and attention. I'd also like to thank the organizers and sponsors for this opportunity to share some information and to share what we've been learning about the pediatric microbiome. This is, as I said, I think this is a field rich with opportunity, and I think we'll be seeing a lot more out of this field in the coming years. So definitely please keep an eye on this space, and thanks everyone for your time.
Thank you, Dr. Hollister. I would also like to thank our sponsor, Roche Diagnostics, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February 2016. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.